spearhead this wonderful initiative from National Institute of Health, and of course, from our partner institutions from the T32 programs throughout the nation. And today, we are going to start with one of these T32 programs that is located in the University of Pennsylvania, or UPenn, endearing way to call it. And the way we're going to do this is the following. The format of this seminar is that you're going to have first our invited speaker, and today is Dr. Amelia Eisch. And after she talks about great science that she's doing, we will have a short Q&A about the science that she presented, followed by the intervention and support from Dr. Arnaldo Diaz, who is Assistant Dean of Research Training Programs at UPenn, where he'll talk a lot about the programs that are available at Penn. The important thing here for everyone, and this is more of a housekeeping kind of thing, is that please, if you want to be part of this series and you want to get an invite, I would encourage you to share your email address with us through the chat. It's just a matter of putting your email address and we'll take care of sending you an invite. And this is an all-inclusive, diversity, welcoming event. So if you want to share it with everyone that you think will benefit from participating in this seminar, please do so, because we have it available for you not only live through live streaming, but also we'll be taping it, so it will be part of our Neuro-ID program webpage. So for those of you who were not able to attend, you can go and tell everyone that they can see it later in our website. So we really encourage you to be part of this wonderful initiative. And I really want to thank our partners and also my esteemed colleagues from the BPN Doer programs that through the summer, where every Wednesday came to join us in this effort, and today is not the exception. So I really appreciate you all here. And also I want to acknowledge, of course, Dr. Jose Garcia Raraz. Jose Enrique, if you want to say some words, just briefly.
Welcome, everybody. I have a terrible connection. I think it's going to fall any moment, but I'm here and I welcome you also.
Thank you, Dr. Garcia. So without further ado, I want to introduce our guest speaker, which is Dr. Amelia Eisch, who is Professor of Anesthesiology and Critical Care and Neuroscience at UPenn, Perelman School of Medicine, and the Children's Hospital in Philadelphia. Dr. Eisch received her bachelor's degrees in science, or in arts, I should say, in psychology and biology from Yale University, and later she continued her PhD at the University of California, Irvine. And after her PhD at Irvine, she returned to her alma mater at Yale to work with Dr. Eric Nestler, specifically trying to examine very much so the role of the hippocampus in the motivation and rewarding processes. And through that time, she discovered a relationship among experimental delivered and self-administered opiates, dentate gyrus, neurogenesis, and the hypothalamic pituitary adrenal stress axis. In fact, a lot of the work she did is something that I discussed in my biological psychology course, Amelia. I wanted to share that with you. So I spread the word of the wonderful data that you collected there. So after her postdoc, she went to UT Southwestern in a faculty position there, and where she probed genetic and circuit mechanisms of underlying behaviors using transgenic mice lines. And in 2011, Dr. Eisch was selected as the inaugural Seymour Benzer Lecturer by the National Academy of Science. And her lab, after being at UT Southwestern, she moved to Penn, And since then, her lab at Penn has continued to be very interested in the hippocampus, but through funding from NIH and NASA, has explored the hippocampal structure and function as it relates to normal and pathological brain function, with a particular emphasis on adult neurogenesis, addiction, and stress-induced depression. And most importantly, Dr. Eisch has documented commitment to training the next generation of neuroscience and underrepresented students from backgrounds in her lab. And she has mentored more than 100 trainees and been PI for the NIH T32 training grants. She's co-director of admissions at the Neuroscience Graduate Group at Penn. And in 2019, she received the highly coveted Distinguished Mentor Award from Penn's biomedical postdoctoral program. So, Amelia, thank you so much for being here, and I will just leave you to talk about this wonderful work you do at University of Pennsylvania. Thank you, Amelia.
Thank you so much, Dr. Maldonado-Vlar. How is my sound? Okay?
Sounds great. Sounds great. Thank you.
Great. So, hello, everyone. I cannot see you, but I can see the chat. So as I talk, if you want to put questions into the chat, I or my fantastic hosts here can check the talk and check the questions. And if it's something of a clarification notice, I am delighted to answer it right away. If it's a discussion point, we can save it and then discuss it at the end. So please use your fingers in the chat. That would be great to see questions coming through. So I'm Amelia. I'm really honored to be the first VP Endure Speaker of the Year. And I'm really stimulated to talk to you about this question. Can a neural circuit of memory be harnessed to combat depression? Very importantly, in mice. And that in mouse part is very important for my work. My lab actually is funded to do 3 different projects that I want to mention. One is from the National Institute of Drug Addiction, and this is to study the role of new neurons and the dentate gyrus in addiction. We're funded from the National Institute on Mental Health to look at whether stimulation of a brain circuit that I'm going to teach you about today can induce antidepressive-like behavior in mice and in rats. And finally, we are funded from NASA to look at the influence of space radiation on cognition. Before I focus in on the middle point, our funding from the National Institute on Mental Health, for the focus of today's talk, I want to briefly mention why these other 2 projects are important, specifically today. And that is that there are 2 students from the BP Endure and Neuro-ID program that I think are on the call who were part of my team this summer. Paula Munoz-Rodriguez was involved with our NIDA funded work, and Jason Lara Jimenez was involved with our NASA funded work. What's really notable about their tenure in our lab this summer is that I have never met them in person. We had only a virtual experience, and from my side at least, it was not only productive, but wonderful. Thank you, both Paula and Jason, for being part of my lab. The focus of my talk today, this NIMH funded work on whether stimulation of a specific brain circuit can induce antidepressive-like behavior in mice, also has a connection, not quite to a BP Endure, but certainly to some of Penn's programs that support fellows from many different backgrounds. Yvonne Soler, who's now in graduate school at Mount Sinai, was both a Penn SUIP and a Penn Prep fellow, and Arnaldo Diaz, who will be speaking after me about the Penn graduate programs, likely will also briefly mention the SUIP and Prep programs, which are summer programs and a post-bac program. So it might be of interest to some of you. Before I actually focus on the data that I'm going to talk about today, I need to give a shout out to who I call my lab partner. And this is Dr. Sang-Hee Yoon, who's pictured here. Sang-hee is a faculty member in my lab at Penn and CHOP, and she's really the head of team stimulation, as we call it. So many of the ideas and data that I'm going to talk about today were really developed hand in hand with Sang-hee. And in some cases, Sang-hee was leading me. I'm a real believer in team science. Now, I really want to focus on the science, but there's two things I need to do before I focus in on this particular question of if a neural circuit that's involved with memory can be harnessed to combat depression in mice. And what I need to do, as many of you students maybe have done all summer, is I need to take this introductory slide and I need to zoom out and I need to zoom in. What I mean by zoom out is I want to zoom out and show you a little bit of what the Eisch lab in summer of 2020 was like this year. This is a picture of one of our sort of Zoom calls and one of our last lab meetings together. And of the 9 summer students that are on here, I've only met 2 of them in person. The other 7 I have never met, and we just had a virtual experience. So I think you can appreciate Paula here and Sang-hee, who I've mentioned, and you know me already. I feel like we're friends already. You can also see Jason up here. Jason, through no fault of his own, did not have an actual picture, but you can see he is also here. Thank you, Jason, for being there. So all these other folks that are on here contributed a lot of data this summer. Unfortunately, most of the data is too piping hot to discuss. I am, though, going to show you some of our data that is unpublished in regards to the question about depression and this novel brain area. But I did want to show these faces, make you recognize what a unique summer this was, not only for my lab, but I'm sure for all of you. And I also wanted to give you a little bit of a roadmap. This picture of my lab in a Zoom call, you will see 2 additional times during this talk. One time in about 5 minutes, and the next time you see it will be about 5 minutes from the end. So if you need to keep landmarks, just keep looking for this Zoom picture again. Okay, so that's zooming out. Now we're going to get into the heart of the science, and I'm going to zoom in. To zoom in and answer this question, can a neural circuit of memory be harnessed to combat depression in mice? I need to go right to the background. The picture that you're looking at here is a brain structure. This is in mice, and it's a structure called the dentate gyrus. The hot pink cells here are stained with a protein stained with an antibody against the protein double cortin, which labels immature neurons. As Dr. Maldonado-Vlar mentioned in my very nice introduction, thank you for that, a little bit embarrassing, but thank you. We are very interested in new neurons in the adult brain. I'm only going to be talking about neurogenesis in one additional slide later today. So here, draw your attention away, if you can, from the hot pink new neurons in the dentate gyrus. And look at this purple sideways V here. This is in a mouse brain, and here you can see in a schematic of the mouse brain, the sideways V of the dentate gyrus. But humans also have the dentate gyrus. In fact, you're looking at a coronal section here through a human brain, and down here at the base of the brain is the sideways V of the dentate gyrus, which is in a structure called the hippocampus. I cannot see you, but I would like all of you to trust me for a moment and put your hands up, please. Give me 2 V's, pretty please, and cross them. And now put them up to your head, sort of like this. That is your own dentate gyrus, okay? Humans have it, mice have it, rats have it, most mammals have it. And this dentate gyrus, you now have I would say maybe a master's level degree in dentate gyrus anatomy. Anytime you see this sideways V in the talk, feel free to do this to remind yourself, oh yeah, that's the dentate gyrus, I know that. Now you might be asking yourself, well, why do I even care? This dentate gyrus, what does it do? Some of the dentate gyrus functions you may already know, because the dentate gyrus does what the hippocampus does in many cases. For example, the dentate gyrus is involved with memory, Dentate gyrus is involved in mood. This structure in mammals is very responsive to stress and helps regulate whether we are feeling depressed or not depressed. A third function that's going to be important for this talk is the dentate gyrus is important in something called pattern separation. Here, it's taking a circle and a square and separating them. But I thought it would be more meaningful if I actually showed you a real-world example of pattern separation. You ready? I want you to tell me if these are the same or different picture. And I'm going to look at the chat. Who knows? Maybe somebody will tell me same or different. Is anybody going to write in the chat and humor the speaker who's staring at the chat? Oh, look at all these people. Love it, love it. Okay, so Dr. Maldonado Vlar gets the first point because she answered first. But look at all these folks. Great. I've got a lot of sames, and then now I've got some different. It's interesting that the folks that maybe took a little bit more time to look thought that they were different. So let's take a closer look. I'm just going to highlight two things. So here's Paula's wonderful face on the left. If you look here, can you see closely? Her eyes are actually closed here. All right, so that's one of many slightly different differences between these two patterns or pictures. It's also noticeable in this picture that Jason looks exactly the same. He's just a name, again, through no fault of his. But Jason, it's good to know you were consistent. These were two pictures that were taken a millisecond apart, and you can see how challenging it is to tell the difference between two patterns or two pictures. That is something that the dentate gyrus is involved in, pattern separation. Particularly when it's really hard to tell the difference between two patterns, like those two pictures, the dentate gyrus really kicks into gear. If something's really easy to tell apart, maybe you don't use your dentate. So that's another function that the dentate gyrus is involved in, and that's going to be very important at the end of the talk. The fourth thing that I want to mention today that the dentate gyrus is involved in is in reward. So we are very interested in that because we study animal models of addiction. So now the dentate gyrus has all these different functions. That's great. So here's the dentate gyrus up here in the upper left. You've got the human brain in the upper right. These are our four, there are many more, but these are four of the main functions of the dentate gyrus. And what caught the eye of Dr. Yun and myself was that the dentate gyrus doesn't always work well. In fact, in many psychiatric disorders, including depression, these functions are messed up, as indicated by this sort of scribbled up memory, mood, pattern separation, and reward. And that's really important because depression is an extremely challenging disease to treat. It's prevalent in all societies, all socioeconomic classes. And if you are fortunate enough to seek help and get on the highway towards recovery, you are put on many different types of drugs. And some drugs are going to work for you and other drugs may not. And finding enough tools, enough exits or entrances onto the highway of recovery is really important for both psychiatrists and basic neuroscientists like myself. So it's a challenging disorder. There are lots of different ways to treat it. And another really important aspect about depression is that 50% of patients relapse. That is not due to their own willpower. That is due to their biology and the challenges that we as people who think about depression in finding the right treatments for people who have depression. That's really an unacceptable relapse rate. Fortunately, as a basic scientist, we have many tools that we can use to discover new treatments for depression. So some of the tools that we have are these animal models that are relevant to depression. We use animal models of stress, animal models of anxiety, and we also use animal models relevant to many of these different functions of the dentate gyrus. One that you're going to hear about today is social defeat stress, also sometimes called chronic social defeat stress. This is a model where essentially the test mouse, which is this small C57 mouse, lives next door to a bully. And let me just label the bully for you in case you need a reminder. That's the bully mouse. This is a retired breeder from another strain of mouse, and it is not happy that it is a retired breeder, and it's not happy that this mouse lives next door, and every now and then, this mouse actually comes into the bully mouse's space, home space. That's where social defeat happens, where the bully literally gets rough and tumble with the test mouse. This is an ethologically relevant model of stress because we can actually then ask this test mouse later, hey, test mouse, if I put you in a novel open space and I put a mouse that looks like a bully but isn't really a bully, just looks like a bully, will you approach the mouse as normal mice do because they approach each other? Or will you be afraid of the mouse and go backwards? This is a test, this social defeat test is going to be really important in the second piece of data that I'm going to show you, which in fact is just in a few slides. So here we have dentate gyrus function, and we have models like social defeat stress, where we can ask animals, are you depressed? Are you smart? How's your pattern separation? How's your reward function? What's interesting about the messed up dentate gyrus function that people have when they are depressed is that with successful treatment, whether it's psychotherapy, whether it's pharmaceuticals, or a combination of them, the dentate gyrus function actually gets better. So this made Sung-hee, Dr. Yoon, and I sit up and think, okay, how can we normalize the dentate gyrus function in an animal that's depressed? And when we were thinking about this, we read a lot of literature and we decided to go upstream. And what I mean by go upstream is we wanted to figure out what the brain structure is that stimulates the dentate gyrus. And we wanted to go upstream to that structure and try to change the activity of that structure so that the dentate gyrus itself would get better in terms of its doing its job, doing its function. So you're an expert now at the dentate gyrus. I hope you're at home making a little dentate gyrus sign. Yeah, maybe. I can't see you, just trusting that you are, This is a slice through a mouse brain through an area called the entorhinal cortex. People many dozens of years ahead of us figured out that the entorhinal cortex has cell bodies that send their arms, their long processes, and then terminate in the dentate gyrus, in this area called the molecular layer. And what's interesting about this pathway, which is called the perforant pathway, is that it can be tickled to make the dentate gyrus more active or less active. And when you look at the literature of dentate gyrus function and entorhinal cortex to dentate gyrus, this stimulation to tickle or increase the activity of the dentate gyrus, you actually can see that there is some evidence that stimulation of the entorhinal dentate gyrus pathway might improve memory. So let me tell you about that specifically. And in fact, I'm only giving you, I think, 5 citations here. These are 5 citations. There are many more that show stimulation of the entorhinal cortex, not the dentate gyrus, but stimulation of the upstream dentate gyrus, of the upstream entorhinal cortex, improves memory. It improves memory in rodents, and importantly, it improves memory in humans. That really caught our attention. And we thought, wow, if all these people have looked at stimulating the entorhinal cortex and normalizing memory or improving memory, what about mood, pattern separation, reward? So in the paper that we published 2 years ago now in Nature Medicine, we looked at the role of stimulating the entorhinal cortex to the dentate gyrus in both memory and in mood. And we used 2 different approaches. If you want to read about the molecular approach to stimulate the entorhinal cortex, I encourage you to read our paper. Because today I'm just going to focus on the chemogenetic aspect, at least in regards to mood. So chemogenetics is a word that you might know, might not. It's got the word chemical in it. It's got the word genetic in it. What is that? You also some of you may know the term dreads. I'm going to take 30 seconds to teach everybody what dreads are, because it's an approach that neuroscientists use often. If you look at the letters down here, dreads, extra D, don't be dreading that I'm teaching you this. It means designer receptors exclusively activated by designer drugs. A simple way to think about this is it is a lock and a key system. We put a lock into an animal's brain, and then we give the animal or inject the key into the animal, and it goes to the brain and it turns on the lock. And by using this lock and key, we can stimulate or inhibit specific brain cells. I'm going to give you a little bit more detail now for folks who are interested. If that's all you need to know, just remember lock and key, lock and key. The lock is a G protein coupled receptor. It's an acetylcholine receptor that has been modified, so it doesn't really recognize the neurotransmitter acetylcholine, but it does recognize this exogenous key, CNO. So what we did in this paper from 2 years ago, we used chemogenetics, which is essentially just using dreads, to express or make the lock appear in mice. And then we gave the animals CNO to open the lock and then to stimulate particular entorhinal neurons. If you're more of a picture person, I'm going to walk you through this first timeline. Here, at this point, when the animal was 7 weeks of age, we did stereotaxic surgery. Yes, I am sometimes a brain surgeon in mice. And we put either a lock that doesn't work, M-Cherry, or a lock, HM3DQ, into the entorhinal cortex. The cells that are in the entorhinal cortex of these animals, this is a specific transgenic animal, so only some of the cells express the lock, and they extend their processes all the way to the dentate gyrus. So one way of thinking about this is by using dreads in a transgenic mouse model, where the genes have been modified, the GPCR lock, the HM3DQ, allows a specific key, CNO, when we give CNO later at 11 or 15 weeks of the animal's life, to stimulate neurons. If you happen to be a gene jockey on the call, you might know that CAMK2I-CRE is a line in which CRE is specifically driven only in glutamatergic cells. And so what this actually allows us to do is we can make the lock appear only in a subtype, the glutamatergic, the excitatory cells in the entorhinal cortex. And then when we give CNO, we can actually stimulate the dentate gyrus. Lots of fancy words. Let's get into the good stuff here. Did we actually do the surgery right? Yes, because we know that animals in their entorhinal cortex showed cell bodies that were red. That's what we would expect if we put the this virus expressing the lock into here. And then these cell bodies projected all the way to the dentate gyrus, and their terminals make this very, very diffuse synaptic innervation in the outer part of the dentate gyrus. So we're in the right place. We got the cell bodies in the entorhinal cortex. We've got the terminals of these neurons, their fingers from at the end of their arms in the dentate gyrus. And we know that this actually works in terms of if we give the key, if we give the animal CNO in an intraperitoneal injection, in the animals that had a lock that just expressed M-Cherry, that's our control, we didn't see much CFOS, which is an indicator of activity in a cell. But in the animals that had the lock that expressed HM3DQ, these cells putatively fired, and that's why they were FOS positive. And this is a graph showing you in the dentate gyrus that in control animals, we saw this many, quote, CFOS positive active cells, and we saw many more cells in the dentate gyrus of the animals that got the right lock. Okay, so the cells are active. That means when we give the animals, the right animals, CNO, we get more neural activity. Well, that's good. We also know that we get more neurogenesis, more new neurons. For any neurogenesophiles on the call, in the animals that had the HM3DQ lock and were given CNO, we saw many more new neurons in the dentate gyrus, shown here. Now you're just seeing part, maybe part of the dentate gyrus. Okay, more activity, more neurogenesis. Who cares? If it's not behaviorally relevant, my lab tends to lose interest very quickly. I certainly would lose interest very quickly. So to ask whether it was antidepressive, we did the same thing. We inserted the lock using viral genetics into the entorhinal cortex. We had the locks ready to be stimulated by CNO, and then we exposed animals to social defeat stress. A reminder, in case you've forgotten, this is the bully model. These animals live next door to the bully. And when we do social defeat stress, this is a model of depression. One of the measures that we use in social defeat stress is shown on the y-axis of this purposefully blank graph right now. It says interaction time. The data that I'm going to show you on this graph is going to show you if a mouse, a test mouse, lives next to a bully mouse in sensory contact for almost a whole day, in physical contact for 5 minutes a day, does this with 10 different bullies for 10 days, and then on the 11th day, we put the test mouse into a big arena, and we put a mouse that looks like a bully mouse on one side. Will this mouse interact with that CD1 mouse that looks like a bully mouse, but this mouse has actually never met before? Well, that's what a normal mouse would do. They would interact with it. Or would you spend less interaction time with it? Because you might be afraid, you might have generalized that all these CD1 looking mice are all mean. Well, let's first look without any dread talk at all. Let's just look at what happens with stress. So you're looking at a white bar here. These are animals that are controlled. They never got stress, and they got the control lock in their brain. So they're really a control for surgery and a control for handling. So these animals, on average, interact with a bully-like mouse, about 120 seconds. Animals that went through chronic social defeat stress spend significantly less time interacting. So this just shows you that stress makes animals tend to avoid less interaction. What we then did with dreads, we then gave these animals CNO, and we went through chronic social defeat stress, and then we did this social interaction test, and we found that by stimulating the entorhinal cortex, it essentially protected against the stress-induced decrease in interaction. For the sake of time, I'm only showing you this one set of data about this, but what we concluded from these experiments altogether was that stimulation of the entorhinal cortex to the dentate gyrus normalizes this antidepressive-like behavior in mice. And we've done this in glutamatergic cells. I'm not going to show you the data, but we're actively working on whether this can work in another kind of cell, a GABAergic cell. So we have data with memory and mood and 2 different kinds of cells, and we're feeling pretty good that in a mouse, we can make that stressed mouse a little bit smarter when it's stressed and a little bit less avoidant when it's stressed by stimulating the entorhinal cortex. That was really exciting to us. But if this is really a dentate gyrus function, I would also expect there to be an influence on other dentate gyrus functions, pattern separation. So in this last part of the talk, I'm going to show you a little bit of unpublished data, and I'm going to show you this picture, which you've seen already. By telling these 2 pictures apart, you're thinking about whether these patterns separate. Are they different or are they the same? And we've decided, or I told you, that they are indeed different. But this is hard because they are very similar. We do a similar thing with mice. I don't show them pictures of my Zoom calls, but we do put them in this apparatus that's called a touchscreen chamber. I'm going to introduce you to it, and then I'm going to play the movie so you can be active with the mice. So you might want to start popping your popcorn now for the mouse movie. In this touchscreens chamber, there is a wall that has here 1, 2, 3, 4, 5, 6 squares. And I think you can tell that the far left and the far right are lit. On this side is a reward chamber where the animals get strawberry Ensure, which we call strawberry milkshake. So in this mouse touchscreen platform, we can ask the animal, hey, can you tell these 2 squares apart? If we teach you to touch the right square and you get strawberry milkshake, you'll probably do that pretty easily. But if your dentate gyrus is messed up and we put the squares closer together, it's going to be a lot harder. So let's watch this mouse, which happens to be a Mr. Mouse. We also do Miss Mice. Let's watch this mouse do easy pattern separation. It's going to get 1 right and 1 wrong. So that mouse got it right very quickly. It pressed the left, there was a high beep, and it went to the reward chamber. Here this mouse is deciding, okay, I'm not sure, is it the right or is it the left? Wait, why am I not getting my treat? I'm totally anthropomorphizing here. It correctly clicked that one. All right, here comes the hard separation. Now the 2 squares are very, very close together. This is hard, but the animal actually gets it right. It touched the screen, it gets its reward. That animal performed a correct pattern separation. Using this and other tests, we ask the question, if you stimulate the entorhinal cortex dentate gyrus pathway, can you improve pattern separation, dentate gyrus pattern separation in a mouse? We looked at easy, we looked at hard, right? Easy is when they're separate, hard is when they're close together, because when they're closer together, it's harder to tell them apart. And we asked these animals, the stimulation animals, that's what this abbreviation means, do they complete more trials? How's their speed? And how's their accuracy? Because just because they complete more trials doesn't necessarily mean that they're better at pattern separation. Maybe they're, maybe somehow the other mice are motorically slower, for example. So when we look at this, and I'm going to go through these data fairly quickly, but we asked easy and hard, hey, mice in the blue that have your entorhinal dentate gyrus stimulated, wow, you did better on both the easy trials and almost on the hard trials. In regards to, they completed more on both the easy and the hard. So that is a sign that they did have, quote, improved pattern separation. But maybe it was because of speed. They actually were faster, but they were faster, this is time to criteria, they were faster specifically on the hard trials, which is sort of interesting to me, that the harder it got, the faster they got. And we also looked at accuracy. Are these animals more accurate, making fewer errors? In fact, on both the easy and the hard, they are more accurate. These are unpublished data that are a piece of the puzzle that we're putting together now to look at this, to answer this next question of stimulation of entorhinal dentate gyrus pathway. Can it improve pattern separation? We're also looking at this in regards to other things, like in our NASA work at the effects of space radiation on pattern separation. And we're very interested in looking and actively looking at reward in these animals. If you stimulate the entorhinal dentate gyrus, will you be able to improve all 4 of these functions? What we have done here is really raise more questions than we've answered. It's exciting to me because I do think that we've shown in mice that the entorhinal cortex dentate gyrus circuitry in regards to depression might be a road to remission. A way of thinking about that is if you walk away from this talk and you think about this highway that people who are depressed are on, trying to figure out which drug to take, maybe someday in the far future, one of those drugs might be actually a treatment, entorhinal perforant path stimulation. That's the hypothesis that we will keep testing. But in the meantime, there are so many other things that we're doing and questions that we are asking. We're really interested in the molecular neuroanatomy of these connections from the entorhinal cortex to the dentate gyrus. As I mentioned, we're interested in reward. We're framing some of this work in a causative angle as well, not just using DREADS, but using other ways to manipulate or remote control the activity of certain cells in the brain. And we're working actively to bring out the best possible bridges between us as basic scientists and our clinical colleagues. Because if any of you on the call know this field at all, you might know that there are actually quite a few papers that say the opposite is true, that the entorhinal cortex dentate gyrus stimulation does not improve memory in humans. That work is very challenging because most of the work in humans is done in patients who have been diagnosed with epilepsy. So we're hoping to work with people that do external stimulation, like transcranial magnetic stimulation, and that opens a population to folks who are depressed, because TMS is often used in severe cases of depression. So I'm really hoping that this will be the year, okay, maybe early next year, when COVID-19 has died down a little bit, maybe this will be the year that we'll actually be able to bring some of those collaborations to fruition. So I'm going to leave you with this final image that even though I think about this in regards to animal models of disease, addiction, post-traumatic stress disorder, depression, I do think the fundamental questions that we're asking and the data that we're getting will help us understand normal brain function as well. You've seen the last Zoom picture, so I'm not going to show you that, but I'm going to show you slightly different acknowledgement pictures. This is the group that worked most recently on team stimulation and team touchscreen. This is our current lab members, everyone with an asterisk, our undergrads. This is just a sampling of our past undergrads and alumni, some of our wonderful collaborators at CHOP and Penn. It's an extremely collaborative environment, and I really have benefited from many of my collaborators that are listed on here and many that are not. And because this week actually marks the 4-year anniversary of me moving to CHOP and Penn, I wanted to leave you with this picture and us flashing you the dentate gyrus sign, so that if we ever do actually meet in person, maybe you can just go like this, and I'll say, ah, you came to my BP Endure talk. That is the end of my talk. I am really excited to see, ooh, there are questions, this is exciting. So I will officially, I'm going to exit my screen so I'm going to look at my phone so I can see faces. And is it okay if I start answering some of the questions on here, Carmen?
Yes, yes, please, Amelia. Thank you so much. So we have several of the questions in the chat. If you want to address them each one from the start, that would be great.
So I'm going to try your first name, Olamide, asked a spot-on question. It's as if I planted you to ask this question. They asked, this is probably addressed by a set of controls, but is it possible that CNO's reverse metabolism into clozapine interfered with behavioral results? Olamide, am I saying your name correctly?
I'm not sure if you can hear me. It's Olumide.
Olumide, terrific question. Olumide knows something that some of you may not. CNO stands for, I'm just going to say clozapine N-oxide. Clozapine is an antipsychotic drug. It's very useful in medicine. There have been reports that if you give clozapine to an animal, or if you give CNO, this key, remember that I mentioned? If you give that to an animal that does not have the lock, that CNO will break down into clozapine, and then clozapine will be active at dopamine receptors and noradrenergic receptors. So, Olumide, fantastic question. And our answer is here, we do have some behavioral controls in the paper, the Nature Medicine paper. In addition, we have a paper that's in revision that looks at specifically in our mouse models and in behavioral tests that are very important to interpreting depression behaviors in mice. Locomotion, exploration of an open field, anxiety, grooming, all of those, if we just give CNO, a dose of CNO that is actually very, very low relative to the paper that showed that CNO can break down into clozapine. If we give a dose that is behaviorally active at DREADS, we do not see any behavioral activation in animals that do not express the lock. So a short answer is yes, there are controls. The slightly longer is only some of those controls are in that 2018 paper, but the rest of the work will be coming out, we hope, very soon. Thank you, Olumide. Great. All right, next question, Alfredo. The social stress model of depression depends on the ability of the mouse to remember how the bully looks. Alfredo, you are correct. How can you know for sure that the stimulation of the dentate gyrus is affecting their mood and not their memory of the bully? Oh, another terrific one. Another terrific one. So those of you that were maybe really carefully looking at the slide, because I knew this had to be a shorter talk, 40 or under 45 minutes, I actually blocked out one bar of the graph that addresses that a little bit. So I can refer you back to the paper on one aspect, but I want to give you a more meaningful answer now. So one of the ways that I do know, Alfredo, that stimulation of the entorhinal dentate gyrus pathway does improve their memory. So I actually, if it improved their memory to the point where they should be able to tell that animal differently, I think they would go sort of in the same direction. In other words, you're pointing out, isn't it important that it's mood and not necessarily memory? And I would say, one, some controls address that, but two, I am okay with mood and memory being connected. Because in many people with post-traumatic stress disorder, that's exactly what we see. That mood and memory influence each other more than we would imagine. We do have some evidence that these animals have better memory, and if they had better memory, I really would have thought that they would know that's not a bully that I've seen before. This is like a cousin. But it's actually the opposite of what we would expect. So this is a question, as with Olumide's question as well. Yeah, I have my email up here, and I would love to continue this conversation if I'm not answering it to your satisfaction. But controls, and to me, it's the opposite effect of what I would think if their memory were actually improved. Great question. I keep waiting for somebody else to ask a question, but I'm going to ask the question. Catherine Yu, wait, is that Catherine Ubry, maybe? How did they stimulate the entorhinal cortex in humans? TMS, TDCS, and what does time to criteria, the graph axis measure, mean exactly? In the human work, Catherine, it changes. In some studies, for example, some of the ones in humans at Penn, they literally put an electrode into the entorhinal cortex and they stimulate it. In other work, they use the non-invasive approach of transcranial magnetic stimulation. I'm putting it here as if I have a magnet in my hand, or transcranial direct stimulation, which is the TDCS. And then in time to criteria is how long does it take them to reach the criteria to go to the next round? For example, in pattern separation, we actually flip the pattern, we reverse it after they get, let's say, 7 out of 8 criteria right for 2 to 3 days in a row, and then we flip it. So they have to get to a criteria, and if they got to their criteria faster, they actually did it better. Thank you, Catherine. Lauren, I think Lauren's the one that needed to go to class at 6. Maybe we still have time. What were your major findings in your experiment? So I would say the major finding that we had is pretty well summarized in our title, that if we stimulate this particular pathway in the brain that ends up on the dentate gyrus, mice appear less depressed. So that's, I think, a good major finding to walk out of the room with. Avi Tessione asks, given the varying nature of information regarding this topic, how likely do you think it is that the same pathway in mice where the entorhinal cortex stimulation is also present in other mammals or perhaps human? Great question, Avi. I'm going to break it into 2 parts. the first part you say, giving the varying nature regarding this topic, of information regarding this topic. I don't think you meant it this way, but I want to actually embrace that and say, thank goodness there is disagreement in this field. Because if there's disagreement in a scientific field, that means hypotheses are being posed and are being proven and disproven. In other words, they're testing their hypothesis. Just a antenna that I hope any of you that go into science have up is if you are ever working on a project and you expect to see one outcome, and if you don't see that outcome, you will be devastated, I would say go back to your drawing board, because you want to define the experiment in such a way that you will either prove or disprove a hypothesis, not just prove. So I really like that there is so much varying information. One of the reasons I was sad I didn't have more space on the slide to put it is that in the last, I would say last 2 years, year and a half, there have been almost a dozen papers on this in humans and in animals. So the data is coming fast and furious now. So it's going to be really fun to put all that together and see what are the intricacies. Is this a parameter issue? Is this, as Avi is mentioning, a species issue? Are mice and rats somehow different than humans? My feeling is that the answer is no. There will be some differences, but there are a lot of similarities. The entorhinal cortex is the main input into the dentate gyrus in humans. It also has GABAergic cells and glutamatergic cells. It's involved in many of these same behaviors. When it's disrupted, you see disruption of dentate function. So I'm going to do a half full, half empty, Avi, for your question, which is that, yeah, they're different, but to me, they're similar enough that if we ask the right questions, we're actually going to learn something about it. And even by just working on this in mice, although I do think it will be eventually applicable to humans, just by working in mice, we will have made a notable scientific advance. A great example is the green fluorescent protein, GFP, which many of you, I'm sure, have heard about. The person who generated GFP got it from jellyfish and just did it because they were fascinated in this particular fluorescent proteins. And now scientists use GFP all the time for so many different things. And they're even thinking about putting it in humans. Okay, I'm going to speed up some of my questions here. Amanda asks, oh, thank you, Olumide, for your thank you to me. Thank you for this talk. You are welcome, Amanda. Thank you for attending. After stimulation of the entorhinal cortex and perforant pathway, how long does the improvement of memory and pattern stimulation last? You were reviewer on our Nature Medicine paper, Amanda, weren't you? And then the second question is, would a future treatment involve repetitive stimulation in order to ensure recovery of depression symptoms? The shorter answer is, it lasts a while. We don't know the details. And the longer answer is, I don't know. No, yeah, that's a longer answer. I don't know if we would have to have repetitive treatment. Amanda, when we first wrote a grant on this a couple years ago, we actually asked that in particular because the reviewers asked it for our paper. And we thought, that's a great question. You know, do we have to do it forever? Because we gave this key, the CNO, chronically here. And the reviewers didn't like it. So we've put that one in our back pocket for now, and I'm hoping I'll get money from another source to be able to fund that. Because like you, Amanda, that's going to be a critical barrier in long-term treatment of depression in a mouse, and of course, a massive translational barrier as well. So I'm thinking right along with you, Amanda. Okay, so Marimar has put some links into the chat that I would love you all to look at while I answer the last question, which is Viviana's. And I want to, you're welcome, Amanda. I want to remind everyone, my email is up there, probably the last one. The Gmail is easiest to reach me at. I also respond, as Arnaldo Diaz will tell you, I respond very well on Twitter. And Arnaldo's up next, so don't go away. Viviana, you get the last question. Here we go. Is the increased interaction time an effect that only happens while the entorhinal cortex is being stimulated, or is it maintained a day after stimulation? Viviana, if you don't know Amanda, you two should become friends, because that's like essentially, you probably typed it without knowing she was typing that, but that's essentially a similar question. We don't know how long it takes. We do know that one dose, one stimulation will not cause these changes. So there is something about it having to be chronic, and the chronic key. And I'm going to leave you with this concept. In traditional antidepressants, marvelous medical drugs, in those traditional drugs, one dose typically is not effective. It takes a while for these drugs to work. So this chronicity part is very interesting to us, that we don't see it with acute, but we do see it with chronic. But Viviana, I'm going to disappoint you, as I did Amanda. That is work that we still need to figure out. Fantastic questions, folks. Absolutely fantastic.
Yeah.
I am going to pass the baton now, right?
Yes, thank you. Thank you.
I'm going to be here. I'm going to be here to make sure Arnaldo is
thank you.
Honest.
Yeah, we need to have, Arnaldo has a presentation, and what I want to inform everyone is that the research part of our talk has concluded. So for those of you who have to leave because of other, important or not so important matters, you can say goodbye and then return next week when we will have our next speaker. That it will be Dr. Andrea Gomez, Assistant Professor of Neurobiology from the Department of Molecular and Cell Biology from UC Berkeley. So she will be talking about her research next Wednesday. But now, the other part of our seminar, which of course is about this networking, is to have the admissions and recruiters to talk briefly about the program. So students that would like to stay to hear more about UPenn, Amelia and Arnaldo are here to present you the wonderful things that UPenn have to offer. So I'll leave Arnaldo to present, and those of you who need to leave, well, thank you for coming, and I hope to see you next week. And please, before you go, write your email address so we could send you our invitation for the next set of series. Thank you so much. So I'll leave it to Arnaldo. Arnaldo?
We can hear you, Arnaldo.
We can see it, Arnaldo. But I don't hear you. You are on mute. Now, let's see.
Now you see me? Hear me?
We hear you, yes.
Thank you. First, thank you, Carmen, Maldonado Blar, for the invitation. Thank you, Araras, and for the opportunity. And thank you, Amelia, for sharing some fantastic research. The reason I'm still working in this field is because of faculty like Amelia and Carmen and Araras that are truly committed to provide a space for our students to grow and to provide a space that is based on respect and understanding, and a space that will challenge our students to be better and to be part of the community. And that's something that I have felt when I was at UPR Rio Piedras and an alum from UPR Rio Piedras, and that's something that I have felt at Penn through the years. So as Carmen said, I'm the Assistant Dean for Research Training Programs and Director of Recruitment and Retentions. I direct the summer program, the post-bac, co-direct a postdoc program. So I work with trainees at all levels. And I'm also part of the faculty in the Department of System Pharmacology and Translational Therapeutics. Throughout my talk, you'll see a lot of pictures of students because I feel the students are the face of the program and the students are why we do this. So Penn, just a nice picture. So you see where we are located. We're in Philadelphia. And even though we're in the city, you can see the city in the back, I mean, the downtown area, you still feel that you have that campus feeling. I mean, the campus is beautiful, the architecture is beautiful, we're surrounded by trees, and it's a campus that is compact in a way that we have 12 schools, and you can see all the schools listed here on the side, and the close proximity of the school, I mean, help in collaborations across departments, across multiple schools. And as if you notice from Amelia's talk, I mean, she lists a few faculty that she collaborates with them at Penn. And that's something that you see in every lab at Penn, in where faculty collaborates across departments within the schools and also across schools. And that's something that is very important for the training, I mean, that our students are receiving. Just to zoom in, as Amelia said, I mean, in some of the parts, this is the School of Medicine portion of the campus, in where you see the Penn Medicine, I mean, buildings. Amelia is in this building right here. But also we are also surrounded by hospitals. Hospital for the Children, I mean, the Children's Hospital of Philadelphia, where Amelia is part. I think her building is right here. But also you see, I mean, that this is a campus that if you see constructions going on, it's a campus that continues to grow. By having new facilities, that means more faculty, that means more training spaces for students. And it's something that in the 10 years that I've been at Penn, I have seen the growth in the number of students that we bring to our programs, and also the growth of the number of faculty that we are hiring at Penn. So the BGS office is Biomedical Graduate Studies programs, is located in the medical school, but we do have faculty across all schools. Just to give a brief, I mean, intro of what BGS is, I mean, this program was established in the 1980, 85 specifically. We have 7 programs, and we had the Biochemistry and Molecular Biophysics program, Genomics and Computational Biology, Immunology, Pharmacology, Epidemiology and Biostatistics, and the Cell Molecular Biology program that have sub-programs. And you can see all of them listed here. And Neuroscience, that's the program that Amelia is the co-chair for admissions. And I've been working with her directly in admissions for Neuroscience, and I'm also involved in admissions across all the programs in BGS. We are a very large, I mean, graduate programs. At the moment, we have 940 students enrolled, in where close to 80% of them are in the PhD track. We have a 16% in the MD PhD, and you can see we have also the BMD PhD programs and a Master in Biostatistics. We have the largest, I mean, I'll talk about the MD PhD later, but we have the largest MD PhD program in the country. You can see the numbers of alum, the completion rate of 86% of our students that start the PhD complete the PhD in our programs, and the time to degrees is around 5.8, I mean, years. In terms of faculty, the faculty at Penn keeps growing. I mean, the faculty that, I mean, are associated with the program. Every time that I give the presentations, I had to update the slide, because, I mean, with more faculty that are in the School of Medicine or the Children's Hospital or at different institutes keep joining our programs. We see that most of the faculty are associated with the medical school, but some of the faculty are associated, I mean, with other schools at Penn, and you can see them listed here across all campus, and also some affiliated institute at the Children's Hospital of Philadelphia, where Amelia, I mean, is part of. But we also have the Wizard Institute, and through the Neuro and the Immunology program, we have a partnership with the NIH. So admissions, the application, I mean, for our PhD programs will be opening now in October, and usually the first week in October. I mean, we're looking for students with strong research experience, and with these students, I mean, that are part of the Endure, I mean, program, are students that know what they're doing, know why they're doing this, and they're getting the training and the mentoring from faculty to be able to apply to the programs and continue their career. So deadline is December 1st. This is, I mean, similar applications as many other schools. I just want to highlight that for Penn, we do ask for a personal statement and we do ask for a research statement. It's 2 separate statements, and other schools will ask you for a statement of purpose that is 1 or 2 pages. And I want to highlight that we do not require the GRE. It's not optional. It's not just not required. And for fee waivers, please, I mean, if you're applying this year, please, I mean, make sure that you write down my email address and you reach out to me for to get the fee waiver code. I mean, so you don't have to pay for the $80 for the application fee. So the grad school career, I mean, it's different on every school. this is, the career path at Penn. Mine at Texas was different, but at Penn, you usually, you have the options of doing a rotation. So our students do 3 rotations. The length of the rotations depends on the program. Sometimes it could be from 10 to 12 weeks. You could do the first rotation in the summer before starting, the semester, the fall semester, or you could just start in the fall semester. During the first year, you take 4 courses. There's some courses that are, similar to all students across, all programs, all 7 programs. And it's usually cell biology. There's a biostatistics course that all of our students take. But then there are some courses that are specific to your program. For example, the students in neuroscience, I'm going to sometimes specify just neuroscience because Amelia, is here, so she can also, answer some of the questions. The neuroscience students will take electrical signals of cell, for example. But the students in immunology will take, of course, immunology. So some core courses are similar across all students, but there's some courses that are specific for each one of the graduate groups. During the first year, you end up your rotations, and then you join the lab. And the second year is when you take your preliminary exam. And every program, graduate program, do it differently. So I encourage you, if you're applying to the schools and want to learn more about how the program is, the exam is structured at a specific program, I'll be happy to set up an individual meeting with you and talk more about the structure of each program. Third year and fourth year, you're in your thesis lab already. You're working, we do support students to go to retreats, seminars, scientific meetings. And year 5 is when you're completing your thesis. As I said before, the time to degrees is around 5.5, 5.8 years. So, there's some additional training that we do offer at Penn, through our graduate programs. There's a certificate in the medical science, program. There's a certificate in public health, and there's also a certificate in environmental health sciences. These 3 certificate programs are closely associated with BGS, but we do have other certificate programs at the university that are also optional for our students to attend. So we have some of our students this year that are doing the certificate in law. So when I started working at Penn, there was only a few certificates, but you can see the number of certificates that have, they're being offered at Penn for students. So for you to complement your training, if that's something that is of interest for you. Something that I didn't mention here, but I would like to mention now is like at Penn in BGS, you are not required to teach. So you're not required to be a teaching assistant. Only the neuroscience program, requires the students to teach for a semester. But if you do like to teach and you want to teach and you want to gain the experience, we will help you find an opportunities, and you will be compensated additionally for that, thus effort. Funding, as of now, BGS is fully committed to the students, and your funding is secured throughout your training. So we provide a full fellowship. The stipend right now is $34,000. We pay for tuitions and fees and health insurance. We also pay 50% of the cost of your dental insurance. We also provide up to $1,000 a year for students to attend conferences or courses outside. The gym is free. It's free membership for the gym, and also we're offering a $1,000 relocation award for all incoming students. I'm a true believer that, in order for our students to be successful, it's not just about paying them and putting them in labs. It's also by providing the right environment and the right support. And that's something that when you're considering grad school, you should, make sure that the space and the place that you're applying is the right fit for you. At Penn, we do have a lot of support for our students. It's a really diverse program. In the past, 4 to 5 years, our, percentage of underrepresented minority students, goes from 26 to 33%. So it's a pretty diverse program. We do offer, career counseling and peer support through the university, through CAPS, the Counseling and Psychological Services program at CAP, but we also have an on-site, a counselor for our students in the medical school. We also, through different student groups, and in this case, CAPSA, we have peer support in where grad students can get advice through, peer-to-peer through, different graduate school students across the university and BGS. We also have several student-led organizations, and I just want to highlight a few. So we have the Ernest E. Just Biomedical Societies that is being established for more than 25 years. And their mission is to provide a space for additional training, professional training, but also to be a support system for our students. Our students, I mean, from the represented backgrounds, I mean, come together as a community and support each other. And I think that's very important as you continue in your career. So we also had a Untitled chapter. This is a newly, I mean, it's probably been in place for 4 to 5 years, in where they have similar, I mean, goals as the EEOS, I mean, chapter. We have the LTVGS, that is called, I mean, it's a support, I mean, for professional and also for support for the LGBTQ plus community. We had the BGS, I mean, this is the Association for Students of the BGS program. We had the Penn Graduate Women in Science and Engineering group that is across the university, and I already mentioned the Graduate and Professional Student Assembly that is more, I mean, across the entire campus. But I mean, in the links that Marie-Marc shared with you all, there's also information about additional student groups. We have student groups that, I mean, work with policy, diplomacy. So it's important that if this is something that you feel is important for you during your training, I totally encourage you to look information at the different schools that you're applying, and there are, I mean, options for you to be part of the communities and also to give back to the community. We have an office, I mean, the BGS Career Development Office, and I also share the link with Marie-Marc. I mean, you can see the Twitter account here. Rebecca Lopez is the coordinator for this office. And there you can find information about core competencies that we as an office, I mean, try to have worked ongoing programming for our students. And you can find information about the different career paths that our alums have, I mean, pursued. You can see that many have gone into research in academia, industry, but you can see that the range of careers in where our alums are, I mean, moving forward. So we also have a career blogging where we constantly publish, I mean, different opportunities for our students, different articles of interest. So I encourage you to look into the different schools that you're applying, and you make sure that, I mean, there's an Office of Career Development that will support you throughout your time at Penn, but will also support you in your transition when you complete your PhD at Penn. So the MSTP program, the Medical Scientist Training Program, as of now, we have 217 MD PhD. This picture was taken during our retreat last year. We have 634 alumni from the program. It's been in place for 62 years. So this is a medical scientist training program fully funded by the NIH. You could see, I mean, we also have 24 BMD PhD students. Last year, 36 students joined the MD PhD program, so it's a really large MD PhD, I mean, program. And this, I mean, is similar. It's the similar career path, I mean, for the MD PhD is similar across all schools. I just want to highlight a few important aspects here. It's a fully integrated program. That means that while you're in the medical school, you're still taking some graduate courses and you do some rotations. You can do one before, I mean, you start first year. You can do also one rotation between year 1 and year 2. But also when you're in the medical school, it's not that you're, I mean, sorry, when you're doing your PhD, it doesn't mean that you're not an MD student anymore. They also have some opportunities for you to maintain engaged with the clinical, I mean, training. And they have a program called Clinical Connections, in where you're paired with physician scientists to learn more about career options for physician scientists, and also, I mean, to maintain, I mean, learning more about the clinical skills needed for a career as a physician scientist. So here you can see something else that I want to highlight is that not all students at Penn MD PhD are in biomedical sciences. You can see that 80% of them are in the biomedical sciences through BGS, but we also have a 20% of the students that are doing either engineering, chemistry, physics, medical anthropology, even business in the healthcare economics program, or history and sociology. So it's a really diverse program also in terms of the backgrounds and the fields in which the students pursue their doctoral degrees. We're looking for academic, I mean, success. Of course, grades, MCAS are important, as well as which courses you took are really important. Research experience, I mean, students coming from Endure programs are really competitive for MSTP programs because they're coming in with strong research experiences and also with an understanding of what it means to be a scientist. We're looking for letters of recommendation from your, I mean, different, I mean, PIs, mentors, I mean, and some clinical and service experiences. So you'll be able to tell us that you're really ready to be a doctor. So I just want to highlight that we'll be hosting a panel next week, September 9 at 7 p.m. And if you follow me on Twitter, you'll see the announcement. If you don't follow me on Twitter, I encourage you to follow me on Twitter. And you'll see, that we have an excellent panel of physicians, scientists. Skip Brass is the program directors, and we have several of our students that are going to be sharing with you insights on how to succeed, as an MD PhD program, but also insights in terms of the application process. So if you have questions about how many shadowing hours you should have before applying, this is a good panel to attend. If you have questions about letters of recommendation, if you have questions about research experiences or taking gap years, all of those questions will be addressed during this panel next week. So my office is the Office of Research and Diversity Trainees, and I'm blessed, to be, with a team of individuals that are truly committed, to the mission of our office. That is not just our recruitment; it's also about providing training and support to our students, especially students from underrepresented backgrounds in the biomedical sciences. I'm the director of the office, the founding director. Lina Ramos, recently joined us. She's the associate director in the office. She's coming in with a PhD in pharmacology and strong, experience as an educator. Najib Brown is the coordinator for the office, but we also have 3 graduate students that are working with us as interns for the office. So we are providing space also for our students to train with us, for those students that have interest in working in program development in higher education. So our office, as I mentioned early on, we have, we oversee programs at the undergraduate level, that's SUIP, the post-bac programs. We provide recruitment and support to PhD and MD PhD students, and I myself, I'm co-director of a postdoctoral program at Penn that is also an NIH program, and where the postdoc, in addition to research, also have a high interest in teaching. SUIP, this is a program that I've been working since 2010. I have hosted 9 Endure, students, from UPR Rio Piedras, including Paula and Jason. I'm very happy by the connections and the relation that I have with UPR Rio Piedras and with Carmen and Araras. And this is a program that initially we were hosting 26 students. In the past few years, we've been hosting 40+ students every summer. And here you can see, as long as I go through the presentation, if I see them, I will highlight those students that were part of the Endure. And I see Giancarlo's here that he's doing a PhD at Hopkins. And I see Jose Gorbea that is at Penn doing a PhD in biochemistry. So the program offers 10 weeks of full-time research. You get exposed to a lot of our faculty. Amelia has been a speaker of our programs, in where you get exposed to the different science going on across, different groups. Career counseling support, we work with the students one-on-one, mentoring support. We do training in responsible conduct of research. We do a lot of social events. So I'm sorry, Jason and Paula, this year was virtual, but we do a lot of social events. We take all students to the Leadership Alliance National Symposiums. And Carmen is also a member of the Leadership Alliance, and we have collaborated, through the years. And then we do as SUIP Research Symposium back at Penn, in where all students interact with our faculty. And here you can see Abdias Diaz, that is also an alum from the Endure, Neuro-ID program. The application will open sometime in the first week in October. It's an online application on official transcript, 2 letters of recommendations, and a personal statement detailing your interest and research interest. Deadline is February 1st. You can see Jose Gorbea also here, along from Neuro-ID, and Leonardo Ramos, that he just started our program, also coming from the Endure program in Puerto Rico. So outcomes for the program, this is, during my time, 2010 to 2019, just before I said the outcomes, we see here Claudia Lopez, also an alum from Endure, and that she's at Penn pursuing her PhD. As I say, 9 students have come from the Endure programs to participate in SUIP. 6 of them are currently pursuing their PhD at Penn. So from 2010 to 2019, you can see that 71% of our alums, you see the M value here, have gone into a PhD or MD PhD, 10% MD, and then you see the different other careers or training program that some of our students has pursued. Some of them are doing postbac, and they're training to become, to apply to PhD or MD PhD, and a few students have gone to other careers. Out of those students, 79 students have come to Penn. I'm very happy and very proud of that. And 10 of them have successfully completed their PhD as of today. in the past couple of months, I mean, few months, some of our alum have secured external funding or have received like awards. And I do keep in touch with all students, regardless which institution you decide to pursue your degrees. And in the past few months, we have received 3 of our alum has received NIH doctoral F31, 1 alum F30, 2 GGN scholar, including, I mean, Tibao Pardo, that is also an alum from the Endure program, 8 NSF, and 3 AAAS fellows. This year, I'm a true believer that is, you know, and this is something that happens when you're in the PhD program, when the life throw you like, you know, some unexpected circumstances, is how you respond to that, what defines you, and how you respond to, is the resiliency, how you respond to those moments is what is going to make you stronger. This year, I mean, due to COVID, I mean, we were, I mean, we were kind of forced to cancel the program, but we were able to provide a virtual experience for our students, and we were able to host 43 virtual summer students, including Jason that is here, and Paula is right here. So, and they were with Amelia. So each student had a research mentor. Each student were, through a virtual course, were able to hear across, I mean, research across different programs with our faculty, a lot of professional development workshops. Students were, I mean, wrote an NSF style proposal. They wrote an abstract. We did a lot of team building activities, mentoring sessions. So it was a really complete program. And I think our students were, at the end of the day, we do this for the students, and we do our best to provide them with the right environment. This, in this way, was virtual, but with the right tools for them to succeed. Quickly, because of the time, the postbac, I mean, we have the postbac since 2004. This is a program that will offer you experience for a year up to 2 years. I mean, after you complete your bachelor's, I mean, the program pays $32,500, and we also pay for your health insurance, and we pay for your relocation, some relocation money as well. And this is some of our students. I mean, Jasmine was just completed the program. She's studying her PhD at Harvard in chemical biology. Elizabeth Mercado, she also completed the program, and she's studying her PhD at Penn as an NSF fellow, and Eric and Kwaku are currently in the program. Online applications, statement, again, similar to the summer program, that the only difference in this application is 3 letters of recommendation instead of 2 letters. Deadline is March 1st. I'll be happy to set up any meetings with all students that, I mean, are interested in any of these programs. The program offers research experience, as I said, professional development, RCR, networking, and community building is similar to the SUIP program, similar to Endure, Mark, Rise. All these programs are in place to provide you with training and support, I mean, to help you transition into a career as a PhD or MD PhD soon. Outcomes from 2004 to 2020, most of our students, close to 80%, have gone into a PhD or MD PhD. As you can see, 8% MD, and others have gone into other careers. I always like to follow my students, I mean, to highlight them and to celebrate their successes. 10 of them have received F31 or F30, 9 NSF, 2 Gilliam Scholars, 1 PD Soros, several diversity supplements, and several are being, I mean, appointed to tutoritors. 27 of our alum have received, I mean, their PhD or MD PhD with a time to degree similar to the time to degree of all BGS PhD students at Penn. Crystal Lenches is with us at Penn. I mean, this is Ivan Soler, an alum from Amelia Eisch as part of the SUIP and also as part of the prep program. He's studying his, I mean, he just started his PhD at Sinai. And Catalina, also an alum from the Eisch lab that is also pursuing a PhD in pharmacology. So that's all the information that I have for you all today. I will open the floor. If you have any questions, I'll be happy to answer them. And here is my contact information, my Twitter account. As Amelia said, I'm really active on Twitter, and the Twitter account for the office, and I'll be happy to take any questions that you might have. And also ask Amelia to join me in answering of the questions if you have any questions.
Well, Amelia has been answering some of them through the chat. She has answered some of them, but I don't know. Amelia, do you think?
Yeah, I can tell you one I did not find yet. Arnaldo, very nice presentation, by the way.
Yes.
Arnaldo, Jason wanted to know what is the applicant success rate of graduate students obtaining fellowships? And I don't know that.
So that's, I mean, Jason, that's a question that I'll be happy to go back to you. I mean, I do know that our students receive support to apply to NSF fellowships. Some students, I'm meeting with some students this week that are putting together, I mean, some application for HHMI. So we do provide support. Several of our students have received F31s. So the success rate, that's something that I can get back to you with a specific answer, but we do support our students through the process of applying, and we do, I mean, make sure that they are putting together and receiving the support while they're applying.
Okay. Yes.
Yeah. Oh, sorry, Carmen. Another one that Melissa was asking if the virtual programs are still open. And I think, Melissa, you might be asking about the SUIP program. The SUIP program is just for the summer. If there was another program that you were interested in, the Penn Prep program, for example, that's accepting applications soon, right, Arnaldo?
Yeah, the application will open soon.
Right, and we don't know if that one will be virtual or not, right?
So I hope I know, because I want to be happy again. But my hope, I mean, we don't know. We cannot predict the future, right? But I mean, what I can tell you is, like, we are committed. We're committed in person. We're committed virtually, and we'll do whatever it takes. And I mean, and I'm surrounded. It's not just me. I'm surrounded by faculty and staff that are truly committed. So we'll run a program. I mean, how it's going to be, I mean, it's hard to say now, but I mean, we will be happy to keep you posted. So we are hosting several events. So our cell molecular biology program will be hosting a retreat, and the retreat will be open to all students. So if some of you want to know, I can send information to Dr. Maldonado, because then you'll have the opportunity to join the retreat, hear about the research, interact with students, and that's something that I can make that available.
Wonderful idea, Arnaldo. Really appreciate that idea, because if the students will have really a good flavor out of what's going on, that would be a wonderful thing. Thank you. Okay, we're coming up to 6:30, and I really wanted to finish at 6:30 because we all are things to do and tired, and with this screen time, you know, it just all day in front of a computer. But I want to finish by thanking Amelia and, of course, Arnaldo, which has always been a collaborator for us here, because he's really a person that is committed to the development of our students. And I want to say this, as always, Arnaldo, thank you.
Oh, thank you.
And Amelia, it was a pleasure to meet you, at least virtually. I hope that maybe at SFN, when we get again in person, I'll find you and I'll give you a hug, and thank you many, many times for what you did today for us and to our students, and really also your commitment and motivation to move forward this needle of inclusive and diversity environments in neuroscience. So thank you so much, Amelia. Okay. And now I guess we'll be saying goodbye for today. And we'll look forward, if you have time,